Okay, so for this audio lecture, we'll be covering information from Chapter 2. Chapter 2 deals with what I call the players, the cells, the organs, and touches upon some of the microenvironment of the immune system, which is needed in order to mature the cells, but also develop them. So first we'll look at the cells and then the organs, and then touch upon microenvironments. So when we look at the cells of the immune system in adult vertebrates, hematopoiesis is the process in which we get our different cells and our different cells of the immune system. In this process, hematopoiesis occurs in the bone marrow. So within the bone marrow, hematopoietic stem cells are going to be constantly renewed, so we can renew the different immune cells. So what happens is the hematopoietic stem cell, HSC, actually gets directed to differentiate into different types of progenitor cells. And two progenitor cells that we'll focus on really throughout the semester is the common myeloid progenitor cell and the common lymphoid progenitor cell. There's a figure that will be coming up that will show you the different lineages that the myeloid progenitors go to versus the lymphoid progenitors. In Chapter 1, we were talking about T cells and B cells and how those are lymphocytes, so you can imagine those are going to be derived from the common lymphoid progenitor cells. So different immune cells are derived from these two different progenitors. So for the immune response, there has to be this coordinated activity between many different cells, organs, and the microenvironment. And again, it all starts with that hematopoietic stem cell that is going to become all these different cells. So we want to keep in mind that all red blood cells and also white blood cells are going to be derived from these through the process of hematopoiesis, and that this is highly regulated. And I would say the holy grail is try to figure out how to push to these different cell types from this stem cell. So, what cytokines, what growth factors would be needed that if we wanted to make a bunch of T cells for someone who doesn't have T cells, how would we do that? So obviously, we don't have all the answers, and this is an area of study of what different factors are needed to push the different lineages. So when I'm talking about the different lineages, this is a nice figure to look at and to think about. So you can see in the middle panel we have our bone marrow, and we start up at the top with a hematopoietic stem cell. Now, again, keep in mind that this will self-renewal, but then it also creates these progenitors. And then these progenitors can go off to different cell types, different lineages. And so if we look at the myeloid progenitor on the left, it will differentiate into a granular site monocyte progenitor, and a xenophil progenitor, and a basophil progenitor. And then we also get our megakaryocytes and erythroid progenitors. Our erythroid progenitors are going to become erythrocytes or red blood cells. So very important. We don't talk about much in the immune system, so you can kind of cut that off there. The megakaryocyte is really important because it creates platelets, and platelets cause clots. And so one of our journal club papers will actually look at how we can get clot formation. And this also becomes really relevant with COVID because individuals, part of the COVID response is that you develop clots because of platelets. So normal response, it helps you from dying from bleeding out, but again, it also allows for the immune cells to get into the location to clear out any kind of pathogen. So platelets are a very important cell. We also have our mast cell that will originate from the myeloid progenitor. Mast cells are a specialized cell that is found in the skin that cause histamine release. So they're actually similar to basophils. Basophils are also known for histamine release. Okay, but basophils come from a progenitor. Xenophils come from the xenophil progenitor, and the xenophil is really good at fighting parasitic worms, so they actually produce a toxin, a chemical that actually paralyzes the worms so that they're not able to attach. So many parasitic worms are intestinal, and so they attach to the intestines through hooks or suckers. And so when xenophils release this chemical, it actually causes those hooks and suckers not to work, and so the worm is no longer attached. And so as other materials move through the GI tract, those worms are shed. Okay, it's the same premise if you like deworm your dog. You're basically giving them like this concentrated amount of this paralyzing substance, not paralyzing to your dog, but paralyzing to those worms in their gut. And then from the granular site, monocyte progenitor, we have a number of different cell types that will be derived. Neutrophils are good at undergoing phagocytosis with bacteria. And then we have monocytes, and monocytes can then differentiate into macrophages or dendritic cells. So monocytes are still kind of naive and young and uneducated, and so they need to go through additional maturation in order to have a function and really an identity, and that's either as a macrophage. Macrophages are really good at undergoing phagocytosis also, and dendritic cells. So there's, macrophage and dendritic cells have slightly different functions, and we'll explore that later. Dendritic cells also can be derived from myeloid progenitors directly, so just keep that in mind. So there's many ways to get dendritic cells. Now, when we look at our right side, we'll also notice that lymphoid progenitors can also derive dendritic cells. So you can imagine dendritic cells are really important for an immune response, for innate immunity, because we have all these different ways in which we can derive them. For the lymphoid progenitor, their primary role, fate, if you will, is creating T cells and B cells. So from the myeloid progenitor, we'll get a T cell progenitor. That T cell progenitor is moved into the thymus, and then will develop into either T helper cells or cytotoxic T cells. And there's actually additional types of T cells than what's shown here, so this is kind of simplifying T cells. But we can see how our T cell progenitor in the thymus ends up developing into our different subtypes. We then have our B cell progenitor, which stays in the bone marrow, but then as it moves out to the peripheral tissue and matures, it becomes a B cell. From the B cell, we can also get plasma cells that secrete antibodies. So there's a different, we can also get memory B cells. So there's different subsets of B cells within that. Another cell type in which the lymphoid progenitor can derive is an innate lymphoid cell, abbreviated ILC. So innate lymphoid cells are derived from the lymphoid progenitor. They're not T cells or B cells, but they have function in the innate system. So they're again, when we think of the lymphoid progenitor, we think, oh, this has a role in adaptive because of the T cells and B cells that it produces. But lymphoid progenitors can produce dendritic cells and these innate lymphoid cells. And dendritic cells and innate lymphoid cells have function in the innate system. So just to kind of keep that in mind. So lots of little exceptions here. So again, try to get the big picture. I would be so happy if you remember that lymphoid progenitor cells derive T cells and B cells, and myeloid progenitors drive our neutrophils, xenophils, basophils, and our platelets. And again, keep in mind that there's additional subsets within this. Okay, and so in this table, we're just looking at the number of cells that are present in human blood. So if we were to take a sample, what would we expect there to be, what percentage, what amount of neutrophils would we expect there to be? And so you can see, if you would focus on the total leukocytes percentage, You can see that neutrophils are the most abundant cell. So if you think about it, we're constantly in contact with bacteria, and we'd want to have a cell that could undergo phagocytosis and engulf those bacteria so that we could signal to the immune system. Okay. Other innate cells like the monocytes, xenophils, basophils, mast cells are at a lower percentage, and they have really specialized functions. Now, when we look at lymphocytes, you'll notice that we have more T cells traditionally than we do B cells, and that kind of makes sense because there's lots of different types of T cells, and so we want to make sure that we have a representation of all those different types of T cells. Where B cells, there are different types of B cells, but there's not as many variations. As far as exam, you're not going to have to regurgitate this. I would definitely want you to know that neutrophils are the most abundant, myeloid cell, innate cell in the blood. So we're going to look more at that common myeloid progenitor and what are the different cell types that develop from it. So again, it's going to be the red blood cells, erythrocytes, really important in transporting oxygen, but there we go. Probably won't mention it again. We have the monocytes, and again, the monocytes can become either dendritic cells or macrophages. And then we have our granulocytes. So granulocytes are called granulocytes because if we look at them under staining, they actually have little granules or dots in them, and that's because of different components they have in vesicles. And so within the granulocytes, we have neutrophils, basophils, and eosinophils. And as I mentioned, neutrophils, they're going to be able to undergo phagocytosis with pathogens, and again, usually bacterial pathogens. Basophils and mast cells have very similar functions because they're going to release histamine, so they're really important in initiation of inflammation and also with allergy response. So if you have allergies, you can blame your basophils. If you get hives, you can blame your mast cells because hives are where you have histamine being released in the skin, and that's because of mast cells degranulating. So, whenever I get hives, I'm always like, curse you, mast cells. Or if I have an allergic reaction, it's like, oh, you basophils. Okay. Eosinophils, these are known for their antiparasitic activity, but they also have antiviral activity. So again, for antiparasitic activity, it's because they release a chemical that paralyzes the worm, so the worm is no longer able to attach. Okay, and then we have our megakaryocytes that are going to become platelets. So just looking at these underneath the microscope, you can see in our first column what they would look like. The middle column is looking at them under SEM, so scanning electron microscopy, and the last column is just a drawing of them in a scientific illustration. So on the first column, you'll notice that the neutrophil doesn't have as much of the punctated staining. If you look at these like freshly isolated, it's a little bit more pronounced, but the granulars tend to be smaller, so it's a little more difficult to see. The staining that's used is usually a Wright stain, WRIGHT. And so that staining has different components in it that will react to the material that's in the granule. And so the color of the granule tells you about the pH of the granules. And so depending upon what components are in there, it's going to react and have a different pH. Okay, and so neutrophils end up having a neutral pH, so hence the name neutrophils. Basophils, you can see those really dark granules, like a dark purple. So those have a basic pH. And then eosinophils are going to have red granules, and that's because they have an acidic pH. And so again, one of the ways I try to remember, like, oh, well, what's the granule? Is just look at the name, neutrophil, basic, basophil, and then eosinophils just have, you just got to remember that's the other one. So they're going to have an acidic pH for their granules, and they have this really red color. So eosinophils are actually really easy to identify. They're very rare, but they're easy to identify because of those red staining to them. Now the neutrophil, because they don't have the granules are smaller, so you don't have those really nice dots on it. One way to identify is through looking at its nucleus, so it's multi-lobed, and so that's kind of unique to the neutrophil. And they're also found at higher numbers, so if you see lots of them, like if you're looking at under the microscope, that would indicate that there were neutrophils. Okay, now basophils look very similar or mast cells look very similar to the basophils in that the granules have that dark purple staining, and that's all that histamine that's in there. Their nucleus is different, so theirs is very circular, where the basophil usually has this like U hook on it, a U shape to it. Okay. So if we look at what's inside the granules, again, you'll notice that with neutrophils, there's a lot of enzymes, proteases that are present. And again, this is so that it can break down and digest the material. There's defensins. Defensins are antimicrobial proteins, so similar to kind of like an antibiotic. And again, it's all so that it can break down the material. And as I mentioned with basophils and mast cells, we've got histamines in there, but we also are going to have cytokines. So when they're activated, cytokines get released, just like with the xenophils, we're going to have cytokine release. But they're also going to have ribonucleases, and this helps with antiviral activity. So for monocytes, what happens is that the monocytes will migrate into tissues and then differentiate into macrophages in the tissues. So this allows you to have specialized macrophages in different tissues. So you have macrophages in your lung, in your liver, and in your skin, and they all, you can kind of think of, they've gone through a special kind of training unique to that tissue. So they're very good at doing their job in that tissue. So they're known as resident macrophages. We also have free macrophages that are in the blood and circulation. So by having these specialized resident macrophages, again, it gives you a better response. It's not that the free macrophages can't respond, it's just they still have a little bit more of a general response. So keep in mind that we don't want to respond to every single microbe that's in our liver or in our lungs or in our on our skin, because some of those are residents too, and some of them are actually symbiotic and beneficial to us. And so by having these residents, they know which is friend, which they shouldn't respond to, versus what they should respond to. So one of the key markings that you'll notice that's different between our monocyte and our macrophages are these pseudopodiums. So these pseudopodiums come off of the macrophage, so we can see how, and they actually are used to engulf material. Dendritic cells also have a pseudopodium. Their pseudopodiums tend to be longer, so if you're to look at them underneath the microscope and they have extremely long pseudopodium, that would indicate it's more likely to be a dendritic cell. So both macrophages and neutrophils are designed and specialized to undergo the process of phagocytosis. So the process of phagocytosis, again, we'll talk about this more so when we talk about innate response, but this is where you're going to have a macrophage or neutrophil that is going to make direct contact with a pathogen and then ingest that pathogen. And so using that pseudopodium, it's going to internalize the pathogen And then it's going to fuse with vesicles that have digestive enzymes so that the pathogen is destroyed in the process. Okay. Now, this digesting of the pathogen is, a good thing because it's going to eliminate the pathogen, right? So if the pathogen is dissolved into all these little pieces, it's no longer a threat. So it's been eliminated in that sense, but it also allows those cells to then present that information, pieces of that pathogen, to our adaptive cells, such as T cells. Okay, so macrophages are really good at presenting to T cells, and they do this through MHC molecules. So macrophages are going to have these surface receptors, MHC, that will interact with the T cell receptor. This process we're going to talk about later on. So again, this is one that, I'm including so you're aware of it, but if you don't understand this process, don't worry about it. You don't have to go read ahead. Again, we'll learn about it in more detail, and I'll have slides and figures that show this process. Okay. Now, with an immature dendritic cell, once it captures antigen, it will then become mature and then migrate to the location. And then it is able to then present to T cells. So again, there's little nuances between the differences in how macrophages and dendritic cells mature that make them unique. Okay. Now, when we look at our common lymphoid progenitor cells, this is our B cells, T cells. We also have natural killer cells, and we also have our innate lymphoid cells, and we can also get dendritic cells from there. Okay, but those B cells and T cells would be the ones that we would recognize the most as being derived from the common lymphoid progenitor. So with lymphocytes, they can look very similar, but they end up having different sets of different clusters of differentiation, or known as CD molecules on the surface. So if we were to look at these cells underneath the microscope, they wouldn't really look different to us. But if we were to do staining with different antibodies that were specific to different clusters of differentiation, these different surface molecules, we would be able to see the difference between them. Okay. And so looking at these next couple slides, this is just going to show you some of the different clusters of differentiation, or CDs, that are present. And you can see on our columns, we have our B cells, our T helper cells, our cytotoxic T cells, and our natural killer cells. And you can see that there are unique CD patterns that would then let us identify which type of cell we have, right? And so some of them are not unique to a single type, but having multiple ones present would be unique. Okay, so with all of these lymphocytes, if we had a sample of cells and we were trying to determine whether these were lymphocytes, we could use this CD5 marker, right? Because the CD5 marker is found on all of these lymphocytes. Now, if we wanted a marker that was specific to natural killer cells, then that's where we could use our CD16. Okay. CD3 is found on T cells. So it wouldn't allow us to differentiate between our T helper cells and our cytotoxic T cells, but it would allow us to know that we're looking at T cells. Now the CD4 marker, that one is specific to T helper cells. So if we wanted to exclude the other lymphocytes and only look at T helper cells, we could stain and sort those cells by the CD4 marker. So this is just some there are additional ones, so there's a second part to this table. So again, a lot of these ones are B cell markers, but some of them are unique to the natural killer cells, and other ones are found on our T cells. So again, coming up with usually 2 or 3 different markers allows us to then look at a specific pool, a specific type of lymphocyte. So when we look at these lymphocytes underneath the microscope, there are subtle differences between them. So panel A is, your lymphocyte that's undifferentiated. So one of the things you'll notice is that the nucleus goes right up, almost up to the plasma membrane, to the cell membrane. And so there's very little cytoplasmic. It's what's known as the cytoplasmic ring. So that's one way to differentiate a lymphocyte from your other cells in your blood. You'll notice with our plasma cell that nucleus has kind of condensed, so there's more cytoplasmic material. And if we think about a plasma cells are producing lots of antibodies, and so we want to make sure that we have a really developed ER to undergo all that processing and folding to produce our antibodies. And then you'll notice with the innate natural killer cells, this one that they're showing, or the innate lymphoid cells, the one they're showing is the natural killer cell. You can see that there's more cytoplasmic material there. It's also a lighter staining color. Okay. Now, if we focus on the illustrations of the cells, you'll notice that, what's unique with our T cells versus B cells is the receptor. So T cells are going to have T cell receptors, TCRs, and B cells are going to have B cell receptors, BCRs. Between our helper and cytotoxic cell, you'll notice that there is an additional molecule. So our T helper cells will have a CD4 molecule, which we saw in our table before, where our cytotoxic T cells would have a CD8, which is unique to it. These, I'm going to call them accessory molecules, help in signaling. So later on in the semester, we'll talk about the signals that are needed in order to activate a T cell. And so the CD4 and CD8 molecule help to stabilize those interactions so that we can get the necessary signaling pathways to occur. So just taking a little closer look at these receptors, the lymphocyte receptors, we can see on our left panel here our B cell receptor. So this B cell receptor has the same structure as an antibody. So when we talk about antibodies later on, we'll talk about how they have our 2 heavy chains and 2 light chains. And so that same structure holds with a B cell receptor. So a plasma cell will actually secrete the B cell receptor, the antibody. Okay, so what happens is the B cell receptors will actually aggregate together, and this allows for activation. So in cell bio, when you talked about cell signaling and the moving of receptors and coupling of receptors, that's what we were talking about. So some of that stuff will come back to haunt you or help you. And then on panel B, what we're looking at is the T cell receptor. So you can see how the T cell receptor has those transmembrane domains that anchor it. It's a dimer that's formed, and so that we end up having our peptide or our antigen being held by the T cell receptor. And as you can see, that CD4 molecule is coming in and stabilizing these interactions because it's holding the MHC, which is also a dimer, also has transmembrane portion. This is class 2, so class 2 is a dimer. It has 2 portions. And so these interactions, if that peptide fits in the peptide groove for the T cell receptor, and we get that initiation of the CD4, we end up getting activation of the T cell so that this T cell will then function. All right. So for the next part of the lecture, what we're going to be looking at is lymphoid organs. And so we can break lymphoid organs into two groups. There's the primary lymphoid organs and our secondary lymphoid organs. Our primary is where our immune cells are going to originate from. So when they go from stem cell to progenitor. So for most, this is the bone marrow. For T cells enter into the thymus as being naive and haven't undergone selection. So the thymus is also. So keep in mind, it's not the thyroid, it's the thymus. So T cells, thymus, bone marrows go through the selection, or B cells go through the selection process in the bone marrow. So think B cell, B, bone marrow, T cells, T, thymus, but not thyroid. Okay, so it's not just any T organ. For our secondary lymphoid organs, this is where the immune cells are going to move to interact with antigen. So this could be the lymph nodes, spleens. There's also tissue called MALT, mucosa-associated lymphoid tissue. There is also, sometimes it's referred to as GALT, so that's gut-associated lymphoid tissue. And there's also some other kind of loosely organized structures, tissues that have lymphoid function. So for the primary lymphoid organs, this is where the immune cells are going to develop. And so we want to keep in mind that during embryogenesis and fetal development, blood cell formation, both red blood cell and also our leukocytes, our white blood cells, are going to shift from sites. So it begins in the yolk sac, but then as the fetus develops, it's going to occur in the kidney. And then that can further develop in the placenta and fetal liver. And eventually what happens is these hematopoietic stem cells will go into the bone marrow late in the fetal development. And then from then on, that's going to be where the population occurs. So this figure just illustrates what I was just talking about. So again, early in embryonic development, it's going to be in the placenta and yolk sac where we have our HSCs. And then once the liver has developed, your HSCs are then going to move into the liver. You're still going to have remnants in the placenta and the yolk sac, but eventually it shifts over to the liver. And then once we have the bone marrow late in development, that's where we're going to find our hematopoietic stem cells. And then after birth, they would be present in the bone marrow. And just to remind you, our hematopoietic stem cell, right in the middle in the bone marrow, that's what's going to differentiate into the progenitor cells and then further differentiate into the different cell types for the immune response in our blood cells, because erythrocytes are important too. Okay, so for the B lymphocytes, they're going to develop in contact with stromal cells that are present in the bone marrow. And so the stromal cells are support cells, so they're going to facilitate proliferation. They're also going to direct migration and stimulate differentiation. We're going to talk more about this process later on in the semester when we talk about B cell development. I just want you to understand that stromal cells are the support cell in the development of B lymphocytes. Okay, and so for our T cells, they're going to initiate from the bone marrow, and then they migrate into the thymus, and that is where they're going to end up maturing and being selected for. And it's in the microenvironment of the cortex and medulla that this occurs. And so if you look at the bottom panel, you can see how we have our blue cell, which is our thymocyte, that is going to move in from the medulla and then go up to the cortex and then back down to the medulla as it matures. During this process, there's a both positive and negative selection occurring, and I don't want to go into that right now because we will talk about that later on in the semester. But you can just notice that blue cell changes from being double negative to double positive to single positive. And so we'll talk more about that process, how that occurs. Now in the secondary lymphoid organs, this is where our immune cell is going to be initiated. So this is where lymphocytes are going to encounter antigen. They can become activated if that's their antigen. They undergo clonal expansion. So if you remember from last lecture, clonal expansion is where you're going to make copies. So you have a good T cell. So in that figure, it was T cell 2. So T cell 2 underwent clonal expansions, proliferated, increase in the number, and then they differentiate into effector cells, into the different types of T cells or B cells. Okay, so secondary lymphoid organs, again, lymph node, spleen, MALT, and there's some other ones, including GALT, the gut-associated lymphoid tissue. Okay. So we want to keep in mind that these lymphoid organs are connected with the blood and lymphatic circulation systems. So this allows for kind of surveillance of the blood and also surveillance of that lymph, that fluid that comes back in the lymphatic system. So, just in case you didn't take AMP2 and talk about the lymphatic system. So this is just a diagram showing, if we look at panel C, the interactions between the circulatory, the blood, and our lymphatic system. So you can see the blood in capillary beds, and right near there we have lymphatic capillaries, so that some of that fluid that gets lost is actually picked up by those lymphatic capillaries and then returns through lymphatic vessels. From the lymphatic vessels, if we go up to panel B, they'll move through lymph nodes, and those lymph nodes allow for interactions between any of that material that's in the lymph, the fluid, and immune cells. So you would have dendritic cells, macrophages, neutrophils, and also T cells and B cells present in the lymph nodes, so that they would be all able to interact together to initiate whether there's a response. Because lymph nodes are found throughout the body, material is going to drain to specific lymph nodes. So if you have your pancreas, there is going to be pancreatic lymph nodes. So if you're an immune cell in the pancreatic lymph node and you come across antigen, you know that that fluid came from the pancreas, so you would know that's where you need to go. Okay. So I always feel like the lymphatic system kind of gets glossed over in AMP2, and it's like such an important system, and it's really cool how it works with it being so closely connected. And one thing this figure doesn't show, panel C, is the valves and such that are in the capillaries. So the lymphatic vessels are just as amazing as your circulatory system circulating blood. So for the lymph node and spleen, they're going to be the most organized secondary lymphoid organs. So those tend to be the ones we focus on just because they're so organized, they're easier to study and to look at the interactions and also the microenvironment that's going to be created. And so when we look at the figures coming up in the next slide, one of the things you'll notice is that, there's going to be this cortex, pericortex, medulla. So because it's so organized, there's going to be these different microenvironments that actually get created, so that you can have cells in different areas So the cells in one area would be different than the cells in another area. And also, I mean, at a microenvironment level of like the amount of oxygen present, different nutrients are present, would also create these different microenvironments. Okay. So for macrophages and dendritic cells, those are usually going to be found in the medulla, where some of our other cells like B cells and T cells would be found more towards the cortex. Okay. And so what happens, and again, we'll look at this in our figure, is that we have vesicles that enter into the lymph node and spleen. Those are afferent vesicles, so I remember it as A, entering. It's the beginning of the alphabet. You have to do that first. And then lymphocytes will exit through the efferent vesicle. So E comes after A, so you exit the efferent vesicle. So one of the things you'll notice is that there are more afferent vesicles than efferent, and that is to slow down the flow of the lymph, the fluid coming in, so it gives these cells more time to interact with the material, potential antigen. So in this figure, we can see our lymph node. The structures that you want to focus on is our afferent vesicles. Those are found right up at the top, and you'll notice that we have more afferent vesicles than we do have efferent. So the efferent is towards the bottom of our lymph node. So again, this is so that we have fluid coming in, but because it only has the one exit point, it actually slows down the flow. So it gives the cells that are found in the lymph node more time to interact with that material, because what's coming in could be antigen. It could be a cell that has undergone phagocytosis and is coming to present antigen. These are known as antigen presenting cells. So they're innate cells that just are interacting with our T cells and B cells. So antigen presenting cells will show this antigen to them, and so they want as much time to be able to interact. You'll notice that we have these follicles shown in the light blue towards the cortex. This is where we're going to have a high population of B cells. In that pericortex is where we're going to have our T cells. Okay, and that pericortex comes right up to meet with the follicle with our B cells. So this allows for T cells and B cells to also interact with each other. Okay, so if you look down in the lower panels, you can see the actual tissue and staining and what it would look like if you were to look at it underneath the microscope. So again, these little follicles where they're stained lighter are where we would find our B cells. Around that is where we would have our pericortex to our T cells. One of the things you'll notice is that this structure where we have these follicles and then the area around them is common to our secondary lymphoid organs. Later on we'll look at pilar patches. So pilar patches have very similar appearance to them. And pilar patches are found in the intestinal tract. So when lymphoid cells will actively migrate towards each other, so they will interact with each other. So just as I pointed out, we have the B cells in the follicle, but then we have that pericortex that's coming right up there, up to it, so that T cells can interact. And this would allow for T cells to differentiate. So differentiation into the different effector cells is going to take place in those follicles in the secondary lymphatic organs. So B cells are going to undergo clonal expansion in the germinal centers. CD4 positive T cells will differentiate into helper T cells so that they can assist the B cells to differentiate. CD8 T cells will differentiate into killer or cytotoxic T cells so that they can go off and destroy infected cells, whether those are virally infected cells or go off and find the pathogen to destroy. This is all achieved through the fibroblastic reticular cell conduit. And so this just allows for there to be a scaffolding. And so if we look at this, this really ends up becoming like a matrix that will hold those cells so that they can interact with each other. So as I mentioned, differentiation into effector cells takes place in the follicles of secondary lymphatic organs. So for B cells, this allows them to mature their receptor. So we have antigen affinity, and that's how strongly the B cell receptor can bind to its antigen. So this is going to increase. There can also be class switching that occurs, and again, we'll talk about this later on in the semester, but there's different types of antibodies, and so switching from having like an IgM, hemoglobin M, to IgG can occur. Okay. Also, B cells and T cells will develop into memory cells in these areas also. So memory cells are saved so that they can be used later on. We talked about that in chapter 1, where we have that primary and secondary response, and part of that is development of the memory population. Now when we look at so next we're going to look at the spleen. So the spleen acts as the first line of defense against bloodborne pathogens, and that's because the spleen has so much blood flow going through it. We'll see that in a figure, but it's one of the reasons if you damage your spleen, if your spleen ruptures, you do risk ending up dying and bleeding, having internal bleeding. So you always want to protect your spleen. And some viral infections can cause the spleen to become enlarged, and so obviously you want to make sure you take precautions so that if you do have an enlarged spleen, and especially if you're doing contact sports, that you don't rupture your spleen. So usually if you are infected with one of these viruses, they would tell you to not return to any contact sports for 6 weeks, or sometimes they'll actually do scans of the spleen to make sure that it has returned to its normal size. Those are all just fun facts, not on the exam. So looking at the spleen, we have two different regions. There's the red polyp and the white polyp. The red polyp is where the red blood cells are going to be found, where the white polyp is where the white blood cells are going to be found. In between that, there's the marginal zone that borders the white polyp. So this is a specialized region where we find high concentrations of macrophages and B cells. So we're going to look at our spleen, not our spleen, but spleen here. So in panel B, we can see how we have those different areas. So the spleen has a lot of red polyp areas where we have red blood cells, that we have blood vessels innervating it, and then we have our white polyp that has, you can notice in that panel B, has a different color to it, and then inside that we have our follicle. Okay, so moving down and looking at panel C, which just, because this is an illustration, shows this a little bit better. If we focus in where we have our follicle here, this is where we're going to have our B cell. Then we have that marginal zone that's present. And then we have our white polyp where we have our T cells present, our T cell zone. Okay, so this allows, because all this blood flow is occurring in this area, if there was a pathogen present, it's going to flow through that white polyp, and so that it would allow T cells, B cells, dendritic cells to, just like in the lymph node, have an opportunity to assess whether this is something that needs to be responded to. Another type of secondary lymphoid tissue is that mucosal associated lymphoid tissue, or MALT. So this is an important layer of defense against infection in areas where we have mucosal and epithelial layers. So most epithelial layers are going to also have mucosal layers to them, and so this is where we might see molt being present. And as I've mentioned, this is similar to GALT. GALT is just specific to the gut-associated lymphoid tissue. So some examples of this, and probably what has been studied more so, is pilar patches. So you might remember seeing pilar patches when in A&P, when you looked at different tissues and you looked at the gut. They very much have that follicle-like appearance that we would see like in the lymph node. And so this is where we're going to have a high population of B and T cells in this follicle. And then we also have these dendritic cells and macrophages. And so the dendritic cells and macrophages, in the most cases, kind of go out and sample. So we see this dendritic cell in panel C that's actually squeezing through this epithelial layer and with its dendrite, with its pseudopodium, and reaching out, and it's to sample that bacteria, engulf it, and then it would bring it back into that follicle where it could interact with these T cells and B cells, and then determine whether it needs to actually respond to it. Okay. The other kind of feature that's occurring here in these mucosal layers is that we can have antibodies. So in many cases, plasma cells are going to secrete antibodies. It gets trapped in this mucosal layer that's on top of this epithelial. And so if those antibodies then interact and attach to a pathogen, because it's specific, again, this would signal for dendritic cells to undergo phagocytosis and bring back that material, so that eventually maybe some lymphocytes would actually end up responding. So we see this interepithelial lymphocyte that could be going off and try to squeeze through those epithelial cells so that it can actually attack and destroy the bacteria. So these are really unique tissues, and again, I would say in the last probably 5-10 years, an area of research is how your gut and the microbes in your gut affect your immune response and your overall health. So the summaries for this chapter is that, although blood cell development is necessary for part of the immune response, it's only the first step. So this is only chapter 2, right? But it's really important that you know and understand the different cells, the different players, the different organs that are involved. There's multiple other organs and tissues that allow for those blood cells to interact and then mature so that they can be part of a functional and robust immune response, a proper immune response. And that these interfaces and tissues are very complex and multifaceted. And that's what we have the rest of the semester to learn about. So we will learn about the innate response and how it develops and matures. And then we will also look at the adaptive response, how T cells and B cells develop and mature. Okay, so again, this is just one of those kind of foundation chapters, just to give you an overview of like what's coming.